Hello everyone. Today we are going to see the topic enzyme inhibition. So enzyme inhibition, it is the most important topic which comes under the enzymes unit. So the name itself says which inhibits the enzyme activity. So inhibition is nothing but just any reaction that affects in the form of any of the molecule or substance, it termed as inhibition. So coming to the enzyme inhibition, so the enzyme activity which gets inhibited with some of the molecule or a substance which is called as inhibitor. So in this enzyme inhibition, we are going to see about what are the types of enzyme inhibition and what are the subtypes of enzyme inhibition. So let's go into the topic. So coming to the definition, so enzyme inhibition is nothing but it's the molecule when particularly binds with the enzyme and prevents it from carrying out the chemical reaction. So it just prevents the chemical reaction. So for example, enzyme plus substrate gives enzyme substrate complex which further gives the product. So if suppose the enzyme inhibitor, a molecule binds with any of the enzyme or substrate, it just not carried out to the enzyme substrate complex and hence no product has been released from that chemical reaction. So this is the enzyme inhibition definition. So usually inhibitors which stops the enzyme activity from binding it to the substrate. So as I told already, enzyme, it has to be bind with the substrate and enzyme substrate complex will be formed. After the enzyme substrate complex will be formed, the products has been released in the chemical reaction. So in this, which stops the enzyme binding to the substrate and effectively slow or even stops the chemical reaction. So enzyme inhibition occurs when certain substances called inhibitors which binds and inactivate the enzymes and just stops the chemical reactions. So inhibitors are the compounds that cause the enzymes to lose the activity by either slowing down or stopping the chemical reaction. Yes, some inhibitors cause temporary loss while others cause permanent loss. So for example, in case of any intake of drugs or antibiotics or poisons or antimetabolites, our body's chemical reaction will get stopped due to the intake of these drugs antibodies. So for example, if suppose if we are in illness, we are supposed to take some of the drugs and medications like chloramphenicol, penicillin, ampicillin. So these substances, these inhibitors may cause or stop the chemical reactions that occurs in our body and it affects our metabolism and it leads to the energy disruption. So for example, penicillin, it inhibits the enzyme responsible for constructing the bacterial cell wall, and the ACE enzyme is nothing but the angiotensin converting enzyme. This is the inhibitors which is used to treat the hypertension and the congestive heart failure. When this enzyme is inhibited, there are no more chemical reactions will occur. So coming to the types of enzyme inhibition, there are mainly two types, which is reversible inhibitors and irreversible inhibitors. So the name itself says reversible, in the sense it can be reversed to the normal stage, and irreversible, it can't be reversed to the normal stage. So coming to the definition, in reversible inhibition, the inhibitor can be removed or restores the enzyme activity. So when the inhibitor is removed from that particular place, the normal reaction will take place in the reversible inhibitor and it causes the temporary loss, because if just the inhibitor is removed, it can be carried out the normal chemical reactions normally. So it is the temporary loss of the chemical enzyme activity, and this substances forms the non-covalent bond interaction with the enzyme. So non-covalent bond in the sense it can be of weak bond, so that it can be easily removed from that place and it can be a temporary loss. So coming to the irreversible inhibitor, so you could see the enzyme is permanently inactivated because it is the form of covalent bond interaction. So covalent bond interaction is the strong bond which cannot be separated from that enzymes to the inhibitor. So it causes the permanently inactivated form of enzyme and it just cannot be reversed back. So it is called as irreversible inhibitors. So reversible inhibitors are further classified into 3 types, that is competitive inhibitor, non-competitive inhibitor and uncompetitive inhibitor. So the name itself says all the things. First one, competitive. So coming to the competitive inhibitor,